Hi everybody. I'm Seth, and this summer, like Dr. Chan was saying, I participated in the John and Violet K Research Fellowship, and then my mentor was Dr. Prescott. So before I get started, I just wanted to say thank you to Papa Jack and Dr. Prescott for providing me all the help and funding and any like background information that I needed just over the whole summer. And so this year I did, or over the summer, I did a quorum sensing in cyanobacteria. And so let's just go over what that is really quick. So cyanobacteria is it's a phylum of bacteria that dates back about 3.5 billion years, and they're blue-green algae, and they're autotrophs, which means that they produce their own energy all through photosynthesis. And so quorum sensing is kind of just the way that cells communicate. And so this LuxI gene, which is the blue gene, it allows the cell to produce chemicals, these proteins called AHLs, and this LuxR gene, it allows for the cell to detect those chemicals in the environment. So it's kind of like the ear and the mouth of the cell. And so what these genes do is they produce and they detect AHLs, which is a protein that as I mentioned earlier. So my previous study covered whether cyanobacteria have the genes necessary for quorum sensing or not. So that's what I worked on last year. And I concluded that they have the gene for detecting AHLs, but not the gene for producing them. So what that means is that this LuxI gene, which is kind of like the mouth of the cell, they don't have the gene to talk per se, but they do have the gene to listen. So they have the ear and they can listen to what's going on around them in their environment. And so I guess why this is important is because if you look at this tree, so it's a phylogenetic tree of bacteria. And so if you look at the circled portion, I know you can't really read it, but it's a cyanobacteria. And so as it's kind of towards the center, towards the beginning of the tree, that shows that it's, like I said, one of the older phylums of bacteria. And so this year, my problem was that while potential LuxR genes were identified, it is possible that the Rocker program, which I'll talk about in a little bit, picked up false positives. So what a false positive is it's kind of like the program could have picked up genes that look like LuxRs or resemble LuxRs, but aren't, necessarily aren't actually LuxR genes. So I sought to ensure that they were. So to go about this, I started from scratch, just so I could get a better database and better results. So I started at GenBank. I pulled down genetic sequences, so as you guys might remember from biology, it's like A, G, C, T, from GenBank, which is a big public database that has all these sequences that were available to me. And from there, I put them into FASTA files, and so it's just kind of like a big text file with all the genomes in it, and I ran this program called Rocker. So what Rocker does is it looks inside of these genomes, and it looks inside of the big text file, the FASTA file, and it looks for the LuxR gene that which is what I wanted to find. And so I used Genius. So what Genius is it's this kind of big toolbox that I was able to use to do like alignments and tree building and a bunch of different other stuff that was really helpful to me. And so one of the biggest things that I used Genius Prime for, or Genius, was to translate my sequences from AGCT to protein sequences. So that's like amino acids and stuff like that. So it was just a little bit easier to use programs. So from there I used Interpro. And so what Interpro does is it looks, again, within the LuxR genes, and it looks for domains. So what a domain is it's a well-studied, well-documented portion of a gene or a portion of a protein that codes for certain things or has different qualities, I guess. And so from there, I lined up the protein sequences and then I put them into phylogenetic trees. I'll talk a little bit more about that. So my sequence alignments, what I did was I first aligned the 16S gene, which is kind of like a barcode gene per se, I guess you could say. It allows us to, I guess, measure the molecular clock of a gene or of an organism to see how, I guess, like how it's evolving and stuff like that. And then I also aligned the LuxR gene. So I actually had 228 LuxR genes, and I think it was like 60 16S genes. But I just kind of showed the general, you get like the general idea. And so what the alignment does is it takes those genes and it lines them up so that you can see where the gene varies and where it is kind of the same. So as you notice, the green portions are what we call I guess housekeeping genes. So it's portions of the gene that are well conserved and that don't change much over time. And so one interesting pattern that I noticed while I was doing the alignment is that the GC content, which kind of handles the complexity and the behavior of the gene, varies more at the portions where the gene is more varied. So I'm not too sure what that means. I have to talk to my mentor a little bit more about that, but it's just an interesting pattern that I noticed. And then, so the phylogenetic trees, this one is the one on the left, or your right, is the LuxR gene, and it has 228 LuxR genes, and then the one on your guys' left is the 16S tree. So you can't really read them, but if you were to look closely, the trees look very different, I guess, taxonomy-wise. The names aren't in the same place, the relationships aren't the same. And so what that says, or what that points out is that, because the 16S is a barcoding gene, it means it kind of evolves over a neutral amount of time. There's not really any selective pressure or any outside factors that play into the mutation of this gene. So you could say that the LuxR gene, there is some sort of selective pressure on the gene to, that's kind of forcing it, or not forcing it, but encouraging it to evolve over time. So I guess just to sum everything up really quick, the cyanobacteria, one of the reasons why, or one of the, I guess, things that this points to is that cyanobacteria have a really hard time growing in pure cultures, so that's saying just with just other cyanobacteria and nothing else. But they do have an easier time growing in cultures that have other organisms such as heterotrophs. So that points to some sort of interaction that could be quorum sensing. And the presence of LuxR in old bacteria, which is very important because we see that this LuxR gene has been around since, well, as you can see, 3.5 billion years ago. And that says kind of a lot about the gene itself. And the LuxR tree versus the 16S tree, which I just talked about, is the selective pressure on the LuxR gene, which means that it's not evolving into like a neutral rate. There's some sort of outside interference going on. And then the evolutionary trends, as you saw the housekeeping parts of the gene, which didn't vary very much, and then the redder, or the lower level red portions of the gene, which were genes that I guess mutate and change over time. And then the distribution of LuxR solos was uneven. To sum everything to close, it was kind of some of the cyanobacteria had 10 LuxR genes, while some of them didn't have any at all. And that's a pattern I'd have to look into a little bit more, but that's just something that that we kind of came to a conclusion about. And so I just wanted to say thank you to Papa Jack, Dr. Prescott again, and then Dr. Chan for helping me with the research this year, and then the Los Alamos National Lab for providing me with the rocker model and everything that I used to do this research. Thank you. And then, sorry, really quick. I guess just from this whole thing I learned that I have a really a deeper appreciation for science now. It's a lot easier to take these things for granted, and I really learned how to work hard and how to persist with things when they get difficult. And I learned a lot of biology. Okay, thank you.